I didn't start off being hooked on rare disease research. I did my PhD on goldfish in a department of zoology, and I figured out how they could survive in the complete absence of oxygen, which is really quite an interesting story. They make ethanol. They're like yeast. They're like the yeast of the vertebrate world. And I came to the MNI in 1985, and again, I wasn't particularly working on rare disease, but my colleague, who unfortunately passed away several years ago, George Carpati, got me into rare disease research. So George was one of the world experts in neuromuscular disease, and he had pathology sessions every week, which we'd attend to learn about neuromuscular disease. And I was kind of floundering. I was actually doing NMR. So I was setting up the MRI stuff and the MRS stuff at the neuro. And that really wasn't going anywhere, at least the spectroscopy part of it. The MRI, of course, was going somewhere. And George, at the time around 1990 or so, said, why don't you work on some of the patients that I have? They have apparently a mitochondrial disease. And the first mutations in the mitochondrial genome had just been reported. And I'd never touched DNA, never touched RNA, and never did anything with this, with genetics really, because I'm a kind of biologist at heart, although I liked population genetics when I was a kid. And at that time, a technique called the polymerase chain reaction came out. So I actually hired George's son, who was a medical student here, to see if we could get that thing to work. So we bought 3 little heating blocks, and I'm sure many of you have heard of that reaction. It's basically a way of amplifying DNA, but you have to move it through different temperatures to get the whole thing to work. So George's son did that, and we ran the thing on the gel, and we got a band, and I said, boy, kids can do this stuff, I'm in. So that got me hooked on it. So I have the great privilege of being now the chair of human genetics at McGill, and there are a lot of researchers in our department who work on rare disease. And at the neuro, I'm part of what used to be called the neuromuscular research group, and we now call ourselves the rare neurological disease group, and it's headed by Bernard Bray, who's a brilliant neurologist who's sitting out in the audience at the moment. So the title of my talk is Rare Disease Research and the Proper Practice of Medicine, and you'll see where I got that title in just a minute. So I took this screenshot from Eurodis that has a video, and I'm sure many of you have looked at their videos. They're very, very creative, very moving videos. And actually, one of the women in this video has one of the diseases that we study, and I'll just mention it as I go along. So what characterizes a rare disease? We haven't really talked much about it, and I'm sure everybody in the audience who's interested kind of knows the answer to that. So it's defined as less than 1 in 2,000 people affected. In the US, it's 200,000 or less Americans affected, which ends up being about the same thing. We know that there are about 6,000 odd. The numbers vary between 6,000 and 8,000 rare diseases. So on aggregate, that makes up a lot of people. So millions of people in the world are affected. And they most often have a genetic basis. I'm sure there are some that are caused by environmental poisons, but the vast majority of rare diseases are inherited mostly in a Mendelian fashion, meaning there's a single gene that produces a very strong phenotype, and it produces something that's recognizable, and it has a very high penetrance. So it disrupts some system, and that's why, one of the reasons why rare disease research, I think, is so interesting, because it gives us some kind of a very robust insight into a complex biological system when we dissect it. But only a handful of them are known. So there's 6,000, and there are many eponymous syndromes that have the names of physicians that might have presented them or discovered them, published them in the medical literature, and most of you have never heard of them. So the resources to cope and research into disease mechanisms vary widely. Depends on whether there's a foundation that gets formed by concerned parents or individuals who have no other people who've been affected by the disease. And I myself actually have a rare disease in my family. It's called Huntington's disease. So many of you have heard of Huntington's disease. It's a pretty nasty neurodegenerative disease. It's progressive. It takes you about 20 years to die, and it's basically horrible. But I watched my grandmother die of it. I watched my uncle die of it. I watched my mother die of it. My brother has the disease now. And I think I've probably dodged the bullet, although I've never been tested for the disease. It's a disease you can't do anything about, but everybody knows, almost everybody in this room has probably heard of Huntington's disease. Anybody know why? Anybody heard of Woody Guthrie? Only one person. She's running the genetics counseling program. Anybody heard of Arlo Guthrie? Arlo Guthrie. What was his most famous album? Alice's Restaurant. So Woody Guthrie was a very famous, very influential folk singer in the 20th century in the United States. He wrote about 3,000 songs. He eventually developed Huntington's disease. And that's what brought the disease to public awareness, because he was a famous guy. It's a bit like ALS. You know Lou Gehrig's disease? Lou Gehrig played for the Yankees. And so there are some diseases that became, even though they're rare, Huntington's is about 1 in 10,000, became very well publicized, became very well known because somebody famous, somebody in the public eye had the disease. There are many, many others, hundreds of other diseases that are 1 in 10,000 that nobody ever heard of. And so those people kind of suffer in silence, if you will. There's no foundations for them. They don't know where to go. They're often misdiagnosed. And we always talk about a diagnostic odyssey. And many of the patients that have the diseases that I work on have such a diagnostic odyssey. They went years and years and years and years and were never diagnosed properly. So there are no treatments for the vast majority of them either. So why study rare disease? Why does somebody who worked on goldfish get hooked, a zoologist, a biologist, get hooked on rare disease? Obviously, we want to do something about it. I mean, if you get involved, if you're in a situation or you find yourself in an environment like the Montreal Neurological Institute, for instance, where people are working on neuromuscular disorders, which are all rare, you get attracted to try and work on the biology of the system or to do something to help the patients. But I think there's something much more fundamental about studying rare disease, and everybody who studies rare disease probably knows this quote by heart. So in 1657, William Harvey, who was a British physician who discovered the circulation, received a letter from a Dutch colleague who wanted him to work on a patient who had a rare bladder disease. Harvey was old by that time. He was 79 years old, and he was very sick. He would actually die a few months later. And so he said, I can't really work on this, but he penned these words, and it's worth just repeating them. Nature is nowhere more accustomed to openly display her secret mysteries than in those cases where she shows traces of her workings apart from the beaten path. Nor is there any better way to advance the proper practice of medicine than to give our minds to the discovery of the usual law of nature by careful investigation of rare forms of disease. And you can read the rest of it. So the idea that he had, and this was 350 years ago almost, is that by studying rare disease, you can basically figure out how physiology works, how biochemistry works, how the body works. And I think that's a really strong motivation for discovering the rare forms of disease, because often in complex diseases that have probably many different genetic components, it's very difficult to dissect them. And I could give you a 1-hour lecture on forms of rare disease that have led to insights into more common forms of disease, rare forms of cancer that have given us models to explain all kinds of more common cancers. And one of the best examples I know in the literature is cholesterol. So everybody knows about cholesterol, right? If you have high LDL levels in your blood, it's bad. It's a risk for cardiac disease. If you have high HDL levels, it's good. So how did that come about? It came about by two young guys who in around 1972 were working at UT Southwestern, and they decided to look at familial hypercholesterolemia, which in itself is not all that rare. It's about 1 in 500. It's an autosomal dominant disease, and there are very rare patients who have 2 bad copies of the gene, which was then unknown. This was, of course, pre-genome. Nobody knew. And so these guys were named Brown and Goldstein, Michael Brown and Joe Goldstein. And what they figured out by using those patients that had the rarest form, they had 2 bad copies of the gene. These were 1 in a million, 1 in actually 1.1 million patients. That was the absolute key to figuring out that LDL actually had a receptor on the outside of cells that allowed that to be internalized, that essentially controlled homeostasis of cholesterol in the body. And that led to the development of drugs that inhibited the enzyme that controlled cholesterol. So in the absence of the inhibitor, you would produce a monster amount of cholesterol, like 6-7 times, if you completely lacked the inhibitor. And the enzyme activity of the enzyme that was normally downregulated, if you could get that LDL into the cell, was like 50 times, 100 times higher than normal. So that led to the development of statins. So there are many different causes of high cholesterol in the blood. Not all of them are due to the mutations in that receptor, And so the insight that they got from studying that one, like, ultra-rare disease, 1 in a million patients, led to the development of a treatment for everybody who has high levels of LDL in the blood. And there's many, many more examples of that. So the diseases that we study are mitochondrial diseases. Whoops. They were called initially encephalomyopathies because they mostly affect the brain and the muscle. They were initially thought to be extremely rare, but that's just because in this sort of siloed world of, cardiologists seeing patients with heart disease, neuromuscular patients seeing patients with neuromuscular disease, people didn't put it all together. And when they put it all together, it actually turns out it's about 1 in 5,000 the birth incidences, which is a fairly common, if you will, rare disease. And it's complicated genetically because the disorders come in 2 flavors. And I'm not going to give you any data on any of this really. I'm just going to tell you at the end of it why this is exciting work, I think, and why people should get involved. It's a really great time to get involved in studying rare disease. So some of the mitochondria that you get, all of the mitochondria that you get actually come from your mom. And inside those mitochondria, there's a tiny little genome that codes for a very few genes, 13 genes that are actually essential to produce energy in your body. And so the genetics of that is very complicated. The rest of the genes that are required to produce that energy are encoded in the nuclear genome. They're transported into mitochondria, and that's where all your cellular energy is made. So these things tend to be less severe, the ones that you get from your mother with later onset. And in fact, the woman that's in this picture that I grabbed from the screenshot, her name is Annie. She was diagnosed at age 44 with a disease called Leber's hereditary optic neuropathy. It's a mitochondrial disorder that can be caused by one of 3 different mutations in genes for one of the enzyme complexes in that energy-producing system. And it took her 8 years to get that diagnosis. That disease happens to be a founder effect disease in Quebec. So 2 women brought it over from France. And in collaboration with Bernard, we figured out actually who those 2 women were. They were on the boat of what was called Les Filles du Roi that the King of France sent over to populate the colony in New France. And we used to diagnose, when I was diagnosing these, about 1 of these a week. And so it's actually easy. It wouldn't have taken 8 years. And now it would take like overnight to figure out what the mutation was in that disease. The other ones were much more difficult until the advent of exome sequencing, because at least in Canada and North America and in Europe, a lot of the patients who had the very severe early onset diseases were autosomal recessive, so they had 2 bad copies of a gene, but the pedigrees looked like mom and dad and somebody who was affected who usually died early. So we had to develop special techniques to try and figure out what those genes were. Now the situation has changed quite a lot. So these diseases now, there are hundreds of them known. And in fact, they're not just affecting the brain and muscle, but they can affect all different organ systems. They can present essentially at any age, and they can be either multi-system or affecting just specific organs. And we don't really understand why that is at the moment. And the field's kind of exploding. So how do we tackle this? So I just thought I'd list, and this is my last slide, some of the recent developments or some of the technological advances that have allowed us to, or will allow us to try to figure out what's going on and to match particular genotypes, mutations in particular genes with clinical phenotypes, come up with mechanisms of the disease and maybe find, avenues to treat them. So the first we've talked a lot about, and everybody's heard about this, is the advent of whole genome sequencing, whole exome, and pretty soon whole genome sequencing, which will cost maybe $1,000. I mean, the initial genome that was finished in about 2001 cost $2.7 billion to sequence somebody's genome, and now it's going to cost about $1,000, and it's probably going to come down a little bit more from that. So it's $1,000 costs nothing. And if you think about, diagnosing these things, nowadays the medical community sends off DNA samples to a lot of different companies to do panels of genes. And that costs thousands of dollars in general. And a lot of these patients on these diagnostic odysseys have actually had many of those panels done and still not found the gene. So I think it certainly makes sense for us as a community to use whole genome sequencing, whole exome sequencing, as soon as the lawyers can figure out how to share all that data and make sure everything's good with that, as a kind of frontline diagnosis almost. But you need to have good clinical inputs, you need to have good biochemical inputs to make sure that you kind of know what you're looking for when you do that. But that's changed the whole scope of rare disease research. 10 years ago, it was really difficult to find the genetic basis of rare disorders, and now it's, in quotes, really easy. It's not like easy, but it's much, much easier than it was before. The genes code for mostly, the genes that are affected in rare disease mostly code for proteins. Some of them are non-coding, but I won't talk about those. And so ultimately, once we know the genetic defect, now we have to figure out what's wrong with the protein. So there have been huge advances in the technologies that allow us to look at proteins, mass spectrometry in particular. So we use mass spectrometers in my lab. I don't have one in my lab, but I have one that I can use down the street, almost like we would use pH meters. So now we can look at the variant form of the protein that we think is causing the disease. Most proteins don't act in isolation. They act with other proteins in networks. And we can ask, who is it talking to in that network? And when it's got this missense amino acid, for instance, how does that change the network? Does it not fold properly? Is it interacting with the wrong guys? Is it a short half-life? So we can look at now all those questions that were really not that approachable 10 years ago. And then the advent of so-called iPS cells, so induced pluripotent stem cells, kind of changed the landscape on our ability to look at different kinds of cell types and tissues. So most of the kind of industry standard in the business I'm in are skin fibroblasts. They don't always express the phenotype, and some patients have cardiac disease, brain disease, whatever. So we really like to make those special cells, those particular cells. And Shinya Yamanaka, about a decade ago now, 11 years ago now, figured out a way to reprogram somatic cells into pluripotent stem cells so that we could now put in a dish those cells, differentiate them into whatever we wanted, essentially, and look at what happens in a cardiomyocyte or in a neuron from a patient who had a heart disease or a brain disease. And we wouldn't have to just rely on skin fibroblasts to do it. It turned out that wasn't so straightforward or simple to do because there's a lot of variability from patient to patient. There's a lot of genetic background. And the final thing, I think, which is actually has revolutionized the world is the development of genome editing technologies, particularly the CRISPR-Cas9 system, which was identified in bacteria. So it's basically a bacterial innate immune system that prevents viruses from infecting bacteria. And now we can go in with that tool, which is very cost-effective, very simple, and edit things. So we can think about creating disease models just by editing. We can correct defects and show that that's the gene we're interested in because it restores a phenotype that we might be able to measure in a cell. Or we could go into an iPS cell, create a whole series of mutants in the same gene on an isogenic background and ask, how do those things affect the phenotype of the particular cells that we're going to differentiate them into? And finally, people are thinking, and it's happening, and it will happen sooner than we think, I think, of editing actual defects, specifically in stem cells, for instance, in hematopoietic stem cells, correcting defects in the stem cells and doing bone marrow transplants and correcting the disease. So I think these four kind of advances in technology have really changed the conversation in rare disease research, and I think we have essentially unlimited abilities, really, to study to identify the genetic defects in rare disease, to study them at the level of the protein, to study them in appropriate cells, and to edit the genome in any way we like to look at pathogenic mechanisms. So, thank you very much.